Hello student, I am Dr. Mohammad Nasser Malik. Today I will teach you glycoside containing drug and they are very important for your study. That is, first drug is Liquorice, second is Dioscorea and third one is Digitalis. So this is the disclaimer. All the content material provided here only for the teaching purpose. And in this you study the glycoside containing drug, their chemistry, their property and uses. So a student will learn introduction, definition, cultivation and collection, test of glycoside containing drug, physical and chemical property of the drug and their uses. So first drug in your this is Liquorice. Synonyms is Muleti, Radix, Glyceriza, Liquorice and just Madhu. The biological source of Liquorice consists of peeled and unpeeled stolon roots and stem of Glyceriza glabra belonging to family Leguminosae. Macroscopical character is order is characteristic. Taste is uh, taste is cylindrical. Taste is sweet. Shape is cylindrical. Condition dry occur in the peeled and unpeeled form. Surface is outer surface is yellowish brown with longitudinal wrinkles. Unpeeled and in peeled one or yellow colored with fine longitudinal ridges. In case of stolen, a scar of the buds can be seen. Fractures are coarsely fibrous in the dark region and splintery in the woody region. Fracture surface show long fibered projecting outwards. So student, here you see different types of this is the unpeeled drug, this is the young plants, this is the dried drug, this is the dried drug with the stolons, and this is the powder form of the Liquorice. Okay. So microscopical character, peridone or film cork cells, several layers with tubular cells. The outer layer are filled with reddish brown content and inner few are colorless. Phallogen are indistinct. Phalloderm with 3 to 5 layer immediately below cork. Parenchymatous cell whose corner is thickened with cellulose. Parenchymatous, some cells contain prism calcium oxalate and minimum starch grain. Secondary phloem is readily the fiber bundle alternated with short phloem element of tangentiality with medullary rays. Medullary rays are distinct and by to multiserrate parenchymatous and continuation with those of xylem. Secondary xylem well represented, divided like phloem by large medullary rays at a regular interval. Xylem consists of vessels, fiber, and lignified wood parenchyma. The vessels fibers are lignified wood parenchyma, and vessels fiber are lignified with wood parenchyma. With the vessels which are relatively with white show sclerenchymatous and broaded pitted thickening. Pith are consist of large parenchyma with intercellular space and contain starch grain with absent in roots. Chemical constituent, it contains saponin glycoside, that is glycyrrhizine and glyceratic acid, flavonoid like licoristine, licoricigenine, isoliquiritin and isolicoritinine, coumarin derivative like henerine and amblyferone, bitter principles like glycemarine, asparage like cetosterol, starch, resin and malic acid, uses demulcent and expectorant, tonic, laxative, diuretic and emollient properties, anti-inflammatory agent like spasmolytic agent, cough and peptic ulcer. Substituent varieties and adulterants and adulterant are Manchurian licorice is obtained from Glycera uralensis. Color is chocolate brown. It does not contain glycyrrhizine, the active principle. The common adulterant is white licorice, also called Indian licorice, derived from the roots of Abrus Precatorius family Leguminosae. The root is very toxic due to an alkaloid aberrine and therefore should not be used in place of licorice. The root possesses a peculiar disagreeable odor and bitter acrid flavor, leaving faintly sweet after the taste. Next drug is Dioscorea. Synonyms is yam. Biological source Dioscorea consists of dried tuber of a number of wild and cultivated species of Dioscorea deltoida belonging to family Dioscoreaceae. Basically, it is used as a food material, especially for the muscles gain or as a nutraceuticals. So macroscopical character is color is chestnut brown with recoiling roots, odor is odorless, taste is bitter, shape rhizome or horizontal tuber. Microscopy of the Dioscorea epidermis is absent. Stele is the major part of the drug. Endodermis is pericycle are distinguishable. Isolation of diazoxanine. The tuber are washed first, then dried, sliced and extracted with hot water or 95 ethyl alcohol for several hours. The alcoholic extract is concentrated under the vacuum and the glycoside is precipitated with solvent, ether or by lead acetate. In the case of acid saponin, followed by the hydrolysis and extracted with petroleum ether 40 to 60 degrees Celsius with isolate the diazoxanine. So diazoxanine is the main active constituent of Dioscorea. Chemical constituent, it contains steroidal saponin glycoside, so diazoxanine and its aglycone is diazoxanine, a small quantity of ecogenine, resin, botazoxanine, alcohol, diascorine, sterol, cholesterol, and stigmasterol, p-cetosterol enzyme, sapogenase. 75% of the diasporia is starch, which is bitter in taste. Stridal saponin diazoxanine also present in the range of 4 to 6%. Diazoxanine is the hydrolytic product of the diazine. Diazoxanine is the precursor for the synthesis of many steroidal hormone. So the active constituent basically diazine, aglycone, and the steroidal like saponin diazoxanine uses diazoxanine used as a precursor for the synthesis of many steroidal drugs like corticosteroids, sex hormone, and oral contraceptive. It is also used in ruthenium. It is also in rheumatism. Substituted variety of adulterant like Dioscorea isiculanata, Dioscorea glabra, Dioscorea opuscifolia. These are the substitution varieties. And next drug, and the very important is digitalis. Digitalis basically digitalis leaves and also called foxglove leaf. Biological source dried leaves digitalis purpurea, digitalis of lanata, family Scrofularaceae, dried at temperature below 60 degree. Geographical source, it is cultivated in England, Europe and other part of the India. Microscopic character like color is dark grayish green or in color, taste is bitter, order is slight, size is 10 to 14 cm long, 4 to 20 cm wide, ovate, lanceolate irregularly, serrate and broadly dentate margin, extra fracture like leaves are pubescent, pinnate, venation, leaves are crumpled and broken. So you see this is the fresh green leaves and this is the diagram of the leaves in pictorial view. Cultivation and collection. It is biennial or perennial herb. It is about 1 to 2 millimeter in height. Grow in grow in grow in the altitude of 13 to 300 meter. It requires 20 to 30 degrees Celsius. Rainfall is 30 to 40 centimeters per annum. The seeds of the jellies are small in size, so they are mixed with sand for the sowing. Leaves are collected in when flowers are fully developed. Generally, leaves are collected in early afternoon. Collected leaves are immediately dried at 60 degrees Celsius. Stored in well-closed container. Dried leaves should not contain more than 50% of the moisture. So microscopic studies show anomocytic type stomata. Trichomes are uniserrate, multicellular, bluntly pointed glandular trichome with unicellular stalk. Covering trichomes, calcium oxalate crystals are also present. Sclerenchyma are stress grain are present in endodermis. Parenchyma are present in three different places. They are upper epidermis, lower epidermis, and pericyclic part. So this is the transverse section of the leaves. Here you see the pericycle, covering sheath, phloem, glandular trichome, endodermis, upper epidermis, palisade, each and everything of the cells. Chemical constituent digitalis purpura contain purpura glycoside ABC, digitoxin, digitoxin, sugar or 6-dehydroxylase, L-ramanose, 2,4-deoxyoxide, and methyl ether. A-glycone part in the cardiac glycoside have stride structure that are derived of the cyclopentane, perhydrophenthrin. Cardiololites are found in also the types of digitalis. There are different types of digitalis varieties and they contain primary glycoside, product of their hydrolysis, secondary glycoside like digitalis purpura, digitalis lanata. D-toxin is a carditonic glycoside obtained by digitalis purpura and digitalis lanata. It is the most lipid soluble of the cardiac glycoside used in the therapeutics. The major pharmacokinetic parameter of the D-toxin include complete oral absorption, which which distinguish it from other glycoside. Cardiac glycoside detoxing may be indicated in the patient with impaired renal function. Digoxin is the most widely used for the cardiotonin glycoside and it is obtained from the leaves of Digitalis lanata. It is a highly potent drug and should be handled with exceptional care. Digoxin tablets are 60 to 80% absorbed. Detoxin Detoxing is indicated when the risk of digitalis intoxication is great, since it is relatively short acting and rapidly limited when they are compared with detoxin. Chemical test as for Killarney test at 0.4 ml of glycolic acid and few drops of 5% ferric chloride solution and a little of drug extract. Further at 0.5 ml concentrated sulfuric acid. The formation of blue color is acid layer is confirm the test. Second test is legal test. Dissolve pyridine in the drug extract up to add sodium nitroprusside solution to it and make alkaline pink or red color is produced. Baljet test, it is a section of usually add sodium picrate solution to show yellow or orange color. Raymond reaction in alkaline medium, the M-nitrobenzene reddish purple coloration appear. Serie reaction in alkaline medium with 3,5 dinitrobenzoic acid, purple color is appear. So these are the test of the digitalis. Uses, it is used as a cardiac glycoside or a group of natural product characterized as their specific for myocardial contraction and interventricular condition. In large doses they cause toxicity, cardiac arrest, systole, but in low doses they are the treatment of congestive heart failure. They have diuretic activities in the improvement circulation tend to improve the renal secretion which relieves the edema which is associated with heart failure. Adulterant and substitution variety Verbesum capsule, the primose leaves and the Symphosum officinalis. These are the substitution varieties. These are the references of this lecture. So you see there are references books, some of internet content, some of the research paper also in this. So you have any queries, any question, you can ask me and you are very thankful. I am very thankful for your lecture. Okay, thank you dear.